Now let's talk about the stages of erythropoiesis. Now, whenever we talk about the stages of erythropoiesis or the genesis of the RBCs, we always talk about it in terms of stages. Now, these stages can be termed as the first one being the proerythroblast. Now, proerythroblast is the first in the stages which occur after the formation of a committed progenitor. Now, the first one is the proerythroblast. Now, it is known as the proerythroblast because it still stains basophilic. And because of this, not much hemoglobin has taken place or hasn't been produced yet. And a lot of the are both the nucleus and the nucleoli are still in place, but during this stage we can observe one, the formation of the RBCs, and the start of the disappearance of the nucleoli, not the nucleus but the nucleoli. Now this stage then turns into the basophilic erythroblast. Again, at this stage, they will stain basophilic because not much hemoglobin has taken place. But at this stage, the nucleus has started to disrupt and the nuclei have totally disappeared. Now, at this stage, you can easily differentiate that not only the cells are rapidly dividing, but also they have a very distinct staining to them. They stain highly basophilic on the cytoplasmic side, while they stain extremely eosinophilic on the nuclear side. The next stage is the polychromatophil erythroblast. Now, polychromatophil erythroblast is the one where complete eosinophilic staining takes place. And at this stage, what happens is that hemoglobin or clumps of hemoglobins start to appear and start to give its eosinophilic staining. And nucleus totally starts to disrupt. The nucleus, the nuclear membrane starts to disappear, the chromatin material starts to disappear, the nucleoli have already disappeared, and because of this, the total disruption of the nucleus has taken place, and this then turns into the orthochromatic erythroblast. Now, at this stage, at the orthochromatic stage, there is complete disappearance of nucleus. The disruption, the distortion that has taken place at the earlier stages is now completed. The nucleus is totally gone. What remains is a network or a mesh-like network of various reticular elements, formation of the hemoglobin in the cytoplasm. Now, this reticular network remains in place because of the formation of the residual elements. And it is also worth noting that at this stage, most of the organelles of the cytoplasm also start to disappear. The Golgi apparatus, the endoplasmic reticulum, they all disappear. Now, this orthochromatic erythroblast then turns into a reticulocyte. Now, reticulocyte is what is also known as an immature red blood cell. Now, it is known as the immature red blood cell because it is this reticulocyte which is then transferred from the marrow or the site of the erythropoiesis into the bloodstream. And when it enters the bloodstream, the maturation from the reticulocyte to the biconcave RBC takes place. Now, it is called the reticulocyte because it contains fine element or reticulum inside them, which can be easily viewed both in the marrow and in the blood. Usually, about less than 3% of all RBCs are usually reticulocytes, but if the count of the reticulocytes increases more than this percentage, it usually signifies some kind of problem in the bone marrow. Now, this reticulum, which is the basis of the name of the reticulocyte, comes from the residual elements of the ribosomal RNA. Now, this reticulocyte then, when it enters the bloodstream, matures into a full-fledged biconcave red blood cell. So the reticulum disappears. There is no nucleus. It attains the biconcave disc. So all of the processes which had started from the first stage, the proerythroblast, that is the formation of the hemoglobin and the disappearance of the nucleus, ends up at the mature erythrocyte stage.